Hello, ladies and gentlemen. It's my pleasure to welcome you for this third talk of this series. And this talk will be related to genetic archive, an engineering view of how the genetic archive mechanisms work, how do they evolve, and the wonderful molecular level automation which makes it happen so reliably and so robustly. Okay. So without much ado, let me share my screen. So, the title of today's talk is Operations and Evolution of Genetic Archive. And this is the third talk of the series of 12. In the previous talks, we have talked a little bit on wonderful properties of cytoskeleton, followed by the wonderful properties of biological hardware, that is, proteins, the kind of possibilities and functions which it can result into at molecular level. And the biggest part is that it is encodable and reproducible. So, closely related to proteins is today's topic, that is operations and evolution of genetic archive. Okay. So, if one looks at the cell, so this is a cell, inside there is a nucleus. Okay. This is a bigger view of nucleus. Inside nucleus there is a nucleolus, which is basically storage and synthesizing the rRNAs. It has ribosomal activities so that it binds sugar molecules to RNAs to make them more dynamic. They can apply force, they can do mechanical work, and so on at molecular level. So that is the storehouse of energy, energetics of so-called nuclear mechanisms. And of course, chromatin is spread all over, that is the chromosomes. So this is nucleolus, these are the chromosomes, these are the 23 pair of volumes of archive, if you please. And then, okay, the volume can be opened into this coil of DNA. This is the famous twisted helix, where the three pitches are used for encoding one pitch on the protein chain. So one amino radical is encoded with three pair of nucleotides as represented on DNA. So that is the DNA. Of course, normally it stays in a zipped up condition. For transcription, for reading it, for making copies of the code, it has to be unzipped, and then mRNA is replicated. Now in mRNA, that is stands for messenger RNA. Basically, the T is replaced with U, that is thymine. This one, uracil, replaces the T radical, so that the binding is not so strong, the zipping does not happen, and it can go on as individual strand. Okay. So here you can see that normally cell as well as nucleus has been treated like a jelly filled body organelle. But then you see, it's not anything like jelly filled. It may still show some properties of that, but it has elaborate internal structures and pathways and tunnels, as you can see, which lead to these pores where rRNA comes out, and it has come out here, and then it starts translating whatever code comes on mRNA and produces through tRNA with participation of transfer RNA, the ribosomal RNA produces a protein chain according to the code that is there on DNA. So that is in nutshell the function. Of course, human genetics is represented by 23 pairs of chromosomes based on shape and size. Statistical study has been done. Uh, Fortunately, in this area, in medical sciences, biology, lot of things have been derived from statistical studies through clinical practices, through various kinds of data gathering techniques. So they have tried to correlate statistically if malformed chromosomes, certain chromosomes lead to certain diseases or certain problems. So that is how the genes have been mapped and correlated to be doing some effect, which is detectable through clinical practice and stuff like that. So This entire map of 23 volumes, not the entire map, but it is the human genome mapping is still is a ambitious project which is yet to be completed. But then quite a bit of it has been done, and that is 23 pair of volumes or the archive code. They look as above, and obviously these are like 23 pair of volumes which could be read parallelly, and not just they could be read parallelly, the same volume can be read at number of places. A number of chapters can be simultaneously read also, and transcripts can be made of them parallelly. Okay, so uh, possibly, of course, at multiple genes with each volume at a time, within each volume at a time. That's what we said. And through statistical correlation, specific medical issues have been correlated with some physical attributes or certain medical issues, and they have been mapped. The question is, what is the significance of number 23? If it were 21 or if it were 25, what difference it could have made logically? So, logically, to me, it is just incidental. It is just a function of evolution that we have landed up with this number. Now, the question is something like asking that suppose you had a couple of vertebrae extra, what could have happened to the body? Or suppose it had a couple of vertebrae less. Suppose instead of 32 teeth, you had 30 teeth or you had 35 teeth. What difference it could have made? So, functionally, yes, some small differences it could have made probably, but logically, there is nothing to suggest that it would have drastically changed anything. Okay, because this is a historical trail. So, whatever is in as is condition, as evolved condition. So, that is what it is. So, the map of is called karyotype. This karyotype is for haploid cell, which where half the genetic material is being used. So it's not pair of chromosome; it is just a single chromosome. Its pair is missing. Now haploid cells are the oocytes or the egg cells. So they are ova and sperm clearly. So they carry the half genetic material from father and half from mother, and the two combine and make one complete genetic set having 23 pairs. Okay. Now it is an interesting question that why nature has made it like this, that half of father, half on mother. For example, there could have been 3 or 4 contributors to genetic code. So if there are 3 types of egg cells, when they all three will fuse, when all 4 will fuse, of course, one is that statistically the probability of fusion of 4 different egg cells, different type of egg cells, becomes relatively rare. So of course, procreation does affect statistically. Another thing is that swapping 50% of database is actually giving you the fastest convergence towards evolving the data set and improving it. If there is any improvement to happen, it happens in the fastest way. 50% data is swapped at any time, at every generation. Okay, so that is the slightly mathematical part of it. Of course, I'm not going into mathematics of it. So genes have been mapped through these patterns of dark and white bands as seen in the karyotype, and they have been mapped and they have been numbered in with certain convention. Although uh, logically it doesn't matter to me where a gene resides in which volume, as long as the system which is reading it knows where it is to be referred, as long as it is able to refer and reference it correctly and map it correctly to the physical location on the cell. Okay, it should make no difference whether some gene is in this one or in this one or in this one. Okay, so it's a matter of academic interest. Of course, some differences can be there, as we shall see further. So these are the white bands. Now, white bands and dark bands. Now, dark bands are the pure DNA sections. The white bands are the regions where nuclear enzymes are clinging to the DNA. Okay, so there are some nuclear enzymes which are closely associated with chromatids, and they are actually clinging to the DNA. So that's what is making a difference appearance. Okay, what is the significance of these nuclear enzymes, and what do they do? Well, we'll see. So 23 pair of chromosomes have complete archived code for the full human body. Okay. All cells have same 23 pairs of chromosomes. Statistically, that has been proven. And they have the same code. They have the same karyotype. But different type of cells have different design and functionality, obviously. Okay. So when oncogenesis takes place and cells multiply, and so same 23 pair of chromosomes are passed completely to the daughter cells as legacy, and they are meant to be propagated onward. Okay. So only in a rare case of terminal cells like red blood corpuscles, which do not generate any other daughter cells probably. So they don't need even nucleus. So those cells may not have even nucleus. But otherwise, by far, wherever chain of oncogenesis lies and different kind of cells have to be produced as daughter cells, cell multiplication. So all the 23 pairs, 23 pair of volumes are passed as it is as an archive. Okay, they are passed as an archive. It's not that they are opened and referred in each cell. So the oocytes, said I said, have 23 chromosomes, no pairs, so they are haploid. Okay, and together when they fuse, they form the full 23 pairs. Obviously, not all genes are used for transcription in all cells. So nuclear enzymes are the ones which operate the bookmarks. So these nuclear enzymes are actually fixing the bookmarks. So they tell the DNA where to open, where to unzip, where to transcribe, what is to be read, what is to be just passed as archive in closed condition. Okay. So which sections of the archive are to be referred selectively and in which order, and the remaining shall remain dormant and they will be just passed on to the daughter cell as legacy. So in order to make the DNA, there is of course very huge length associated with the DNA strands which are there in human nuclei. So they have to be compactly wound. So they are wound on molecular bobbins of histone octamer. So histone beads form some kind of a bobbin on which the DNA is wound, and there are several histone beads which form the chromatid. Okay, which form the structure of chromosome. Okay, so if something has to be read, the first thing is the histone bead has to be molten away. So the bobbin melts or shrinks so that the coiled DNA becomes free and it spreads, and then it is unzipped, the two strands separate, and then mRNA starts reading the strands. Okay, and it starts copying the code and carrying it to rRNA through the passages and pathways that you saw in the nucleus structure. And rRNA is actually hooking up a tRNA, which reads the codon, set of three codes, the codon, three nucleotide pitch, and anticodon bearing tRNA comes and latches on to it, and then on successful latching in rRNA wise, uh, it delivers the the amino radical carried by the tRNA into the new form protein chain. So protein chain gets extruded at the fag end of the tRNA into the cytoplasm. So that is the mechanism. Of course, nuclear enzymes melt away or shrink the histone bead. So which histone beads have to be shrunk? That activation has to be done robustly, intelligently, as programmed through the nuclear enzymes. Okay. So they trigger unzipping and they enable the transcription. Okay. So, and after transcription has been done, they initiate the rezipping and they close the chromatid for the cell division. Okay. So This is the complete function. So all the operations on the genetic archive strand are carried out by the nuclear enzymes. Now, one question is, from where do these nuclear enzymes come? Obviously, those two have been coded somewhere on the genetic archive, and they would have been pre-produced already to carry out these functions, and they do reside in nucleus. Okay, which means if you just isolate a chromatid, it is no good. There is nothing you can do with it until it is there in its nucleus, surrounded with the nuclear enzymes that are already there and that are already operating, and they know where to move the bookmark, which sections to open and read, transcript, and subsequently translate at rRNA. Okay. So apart from genes, there are some sections in between, like spacer DNA, so called, where often you find meaningless seeming sequence of TA repeated. Now, what do these meaningless codes mean? They are obviously not genes, because it will not produce anything meaningful that way. So, probably if you apply some sense of engineering, probably the molecule is using them as counting beads, just as Japmala is used for counting how many times you utter Ram 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 or whatever. Yeah. So it is used for counting how many transcripts have been copied. Now, obviously, any data center has to have a control on how many transcripts are to be made. Okay. So probably they are used for counting. So every transcription will probably uh, wind one TA code, okay, one TA sequence. And as you go on making transcripts, the strands having number of these TA, they will go on closing progressively. And when all of them are exhausted, probably rezipping is initiated. Okay. So could be something like that. As an engineer, I am tempted to think of that possibility. Now, rest is, of course, to our biochemistry friends to investigate and prove it or otherwise. But then, from engineering point of view, this looks like an interesting possibility, okay, which needs some exploration. So, alternatively, of course, rezipping could be triggered or enabled under certain conditions on getting signals from some adjacent cells or adjacent media in terms of some protein bearing certain structure, certain code, okay, which is identified as some signal to take some action. Okay. So if the adjacent cell is of different type, or it is exerting too much pressure, probably uh, something changes in the program, and it shifts the bookmarks to the other type of cells probably, and it terminates the ongoing replication of cells, identical cells. Okay. So the participants in the translation process are messenger RNA, okay, which of course contain uracil in place of T. Okay. So they are able to flow as a single strand. They will not immediately bind with their counterpart as strongly as DNAs do. Okay. So with Crick-Watson pair kind of matching. So here it will not happen. So that is there. And this strand finds a way in between the two lobes of ribosomal RNA or rRNA. Okay. So what makes it to navigate to the ribosomal RNA and locate itself, orient itself so that it enters in between the vice? Okay. Now that is a mystery. Okay. But what is known is, of course, this codon is matched with an anticodon which is coming from transfer RNA or tRNA, and at the fag end, this is carrying some amino radical which is matching with this anticodon. So this is what is controlling the genetic code as it stands. Okay, which amino acid means what code here. Okay, and it is quite robust. It is universal everywhere. It is the same code. Okay. So now the question is, what controls the oncogenesis? So during oncogenesis, daughter cells have produced are of different type than mother cell. So bookmarks must be moved accordingly. And who does it? Now all such questions actually traditionally they have been assigned to so-called enzyme, okay, which are named after the process. So we don't know how do they do it actually. Okay. So none other than nuclear enzymes are said to be doing it. Okay. How do they do it is a gray area still. Okay. How do they do it so repeatably, so reliably, without hardly any error? Okay. How do the mother cell knows that the daughter cells would have to be different? And different in what respect on next cell division? And where and how the bookmarks needs to be shifted? Which are the next set of genes which have to be expressed and which genes shall be suppressed hereafter? Now, that is a mystery, that is a gray area. But nuclear enzymes are said to be doing it somehow. uh, But that somehow is very, very robust and it is very, very repeatable. Perhaps this information of oncogenesis, where the bookmarks have to be moved, is also stored elsewhere as a genetic code other than gene. Just like treasure hunt, the game goes on, that it gives some instruction that, okay, you go at some location, okay, then take some action, whatever you find on the chit, then go to some other location, again fish out a chit, and whatever directions are written, do as per that. So the entire genetic coding appears to be something like this. It functions like that. It functions like a treasure hunt. Okay. So some code is generated, it guides you somewhere. When you go there, you get the next instruction, and that's how the game goes on and goes on and on. But it goes on and on quite repeatably, quite reliably. Okay. And with full error proofing. Okay. So that is the wonderful part of it as far as engineering aspect is concerned. Okay. So going by the completeness of code in each cell, any cell should be able to grow into full human body because it has all the information, but that does not happen. Probably owing to predisposition of bookmarks, because ultimately what that cell will do will depend on where its bookmarks are. Okay. Plus, of course, it has to match with the pre-existing structures, including interspaces and the conditions existing in the seed cytoplasm. Now, it is also important to note that with just DNAs, you can't synthesize a cell synthetically, artificially in a lab. It has to have a seed cytoplasm. It has to have a seed cell, which has to have predisposition to do those activities correctly. So when the code matches that predisposition of the seed condition cell, the seed nucleus and the seed cytoplasm, then only the game moves forward. Otherwise, it gets terminated. So, experimentally, it has only been proven that inner intestinal lining cells can only fertilize and go a little bit further as if they were oocytes. Okay, but none of the other cells are able to do even that. Okay, so clearly it is a foolproof mechanism. So unless all the conditions match, unless all the keys match into the keyways, probably it will not function. The locks will not open. So next debate is, of course, why have two strands in DNA? Okay, because only one shall have the code really pertaining to the protein. The other one will be anticodon. Okay. Now anticodon, if it is read as a code, it will mean something totally different. Okay. So it in no way will represent the protein that is to be produced. So DNA will have two complementary strands, of which one is the main gene. Okay. All the functions of gene are attributed to that half strand. The other one is merely the keeper or zipping strand. Obviously, the two have different code sequence. How does mRNA know which one is the real gene and which one is not? Which one needs to be replicated and which one is to be ignored? Now, that's a question from engineering perspective. So it is tempting to believe that probably both may be getting transcripted. So anticodons from the complementary strand, they are transcripted, and that mRNA actually carries the tRNA dangling to it, just like clothes are hung on rope for drying. Okay, I will show you a better illustration on next slide. Okay, with their mRNA in the same order. So basically, the code is coming in some order. So amino radicals are presented in some order, and their anticodons are also coming in the same order. Okay, so one mRNA for the main code goes to the upper lobe of rRNA. Okay, the upper jaw of Weiss, molecular Weiss, that if you please, and the complementary mRNA goes to the lower jaw of Weiss. That presents the chain of anticodons, and with the set of 3 anticodons, there is a tRNA dangling down, okay, which is supposed to fish the appropriate amino radical from cytoplasm. Now that can be probably expected because amino radicals will be very small molecules and they can freely diffuse through the cytosol. So the tRNAs which are empty, they can, or which get empty, they can fish through the cytoplasm and pick up the next available correct amino radical matching with the anticodon that is it has on top. Okay, so and of course there can be number of rRNAs in sequence, and the same mRNAs could be passing through all of them one after another, and there could be parallel proteins produced at number of rRNAs. Okay, so yes, so the thing is, in first rRNA, uh, the amino radicals get consumed. By the time the mRNAs reach the next station, the next rRNA, they would have fished already the amino radical from cytoplasm, and they will reach to the site of next protein synthesis parallelly, and then third one, fourth one, like that it can go on as an automated mass production factory, if you please. Okay, so that looks feasible because tRNA, suppose it loses amino radical, now it has to do two things. One is it has to come and get aligned with the rRNA, the lower lobe of rRNA, so that its anticodon gets juxtaposed to the codon. So it has to jiggle, it has to find orientation, and it has to waste a lot of energy in doing that. It is unlikely that biological systems will waste so much energy. They are highly optimized, much more than engineering systems are, of course. They are at the next level of optimization. So, and they are at next level of molecular level automation. Okay. So the tRNA synthetase kind of entity, that you have another ghost enzyme, a god enzyme, so to say, which is responsible for finding out the matching anticodon with the amino radical and picking up the amino radical on tRNA and then propelling that tRNA, aligning it to the rRNA, and I mean it's too much to expect because these are macromolecules. These will not be tangle free if they diffuse through cytoplasm. Okay, so they have to be clearly guided and aligned. Okay, they cannot be just freely diffusing in cytoplasm, right? As has been assumed so conveniently for so many years. So mRNAs from both the strands may work like automation conveyors. So one gets codon, one conveyor gets codon on top, other gets anticodon at the bottom, rRNA, and the tRNA is dangling to it, and it is picking up, it is fishing the amino radical when it comes to cytoplasm, and then it assembles the protein, and after that It picks up, it fishes the next amino radical from cytoplasm, assembles the next protein, and like that it can go on like a factory, like a mass production protein factory. Now, which is tempting to believe. Okay. So jiggling, picking up, matching, cross matching, orienting, reorienting, hit and trial, all those are not going well with the engineering concept of optimization of energy and movement. Movement consumes maximum energy, by the way. So why there will be so wasteful movements in the cell, consuming so much of oxygen, so much of fuel, so much of sugars, in just jiggling and finding the right match, right? So both the complementary strands of DNA are clearly participating in translation, and they also make the robust and error-proof execution. If codon does not match with anticodon, the game will stop there until it matches. Okay. So it is kind of robust. It is cross-checking. It is cross-verifying. It is error-proofing. All those features automatically come with this redundancy and duplication and cross-matching of codon with anticodon at molecular level during the process. Okay. So the main strand comes in one lobe. Now in this picture, it is shown as the bottom lobe. Okay, picture is upside down. Sorry about that. And the upper one is the one where tRNA is dangling, and it is extruding the protein chain made of 20 beads of 20 different colors as represented here. And these codons and anticodons must match. Only then this thing moves forward, and it sheds out the protein by one pitch and moves over to the next anticodon codon pair. Right now, if you turn it by 180 degrees upside down and equate it with some engineering mechanisms that we see in factories, of course. Now here you can have the main code carried on one conveyor. Okay, so the main codons are carried on one conveyor, which is one mRNA. The anticodons are carried, so complementary strand transcript. Okay, so these are the anticodon carriers, and they will always match and they will always arrive in the same sequence. So there is no hit and trial. It automatically happens. It's like an automated conveyor system. It is getting things in the correct order, in the order that is required. Okay, so no jiggling, no hunting, no aligning, etc. Everything is automated. It comes, it arrives in the correct location, and then it fishes the tRNA dangling down, fishes the appropriate amino radical, and it incorporates in the protein chain. So simple. Okay. So only thing is, yes, here this belongs to this one, and of course this belongs to this one. Okay. So I hope the illustration is quite clear, it's quite strong. It makes a very strong engineering case that molecular level you have code conveyors, the dual code conveyor, which do mass production of proteins without any errors, with cross checking, with cross verification, with proofreading function. Okay, and of course whatever is extruded has to fit into the cavity, the birth cavity, which is there already in cytoplasm. If it mismatches, it can get truncated there also. So there are several levels of security. Okay, just like cyber security has many levels, here also you find the biological cyber security, if you please, has multiple levels at which checks and balances are there. Okay, so there are several mysteries of transcription and translation. That is, DNA has two complementary strands, of which only one is the main gene, the other is the zipping code. Okay, how is it that there is no framing error? You have to move by pitch of 3. Suppose you move by pitch 1 pitch or 2 pitch, the entire code will mean something else. Okay, because the triads will be read differently, but that is not known to happen. So how is the framing error prevented in this kind of data archive is a wonderful concept, of course, but it's not known yet. It has not been researched sufficiently. Okay. So what prevents the reading of code in reverse direction? For example, suppose somebody reads the code ulta. Instead of front to back, you start reading from reverse side. The code will mean something else, obviously. Okay. Now that, of course, our biochemistry people tell us that there is a C5 carbon and there is a C3 side of chain. So one is the forward side, other is the reverse side. So it knows how to align so that it aligns in the correct direction. So okay, we grant it at molecular level. Some mechanism stereo specific structure is there which makes sure that it is to be read in the correct direction always and never in the wrong direction. Transcription never happens in the wrong direction. It always happens in the correct direction, the correct sequence. Next mystery, how does mRNA know where it should move, where how it should navigate, to which RNA it should latch? Okay. And how many such target RNAs are there one after another for the translation of the code? How does mRNA know which way it should go inside the nucleus? Okay, that's a mystery. Of course, there are some pathways which are shown, and the probability that it will find rRNA and then it will enter into the rRNA wise, it is the chances are remote, and that will not be optimal again. What is more likely is that as the mRNAs are excluded out from chromatids, en route they may be picking up rRNAs through nucleolus. So when they arrive, they may be passing through nucleolus, and rRNA may be sitting on top of mRNA, and the whole thing moves out, and rRNA gets lodged into the wall of endoplasmic reticulum, and then it starts synthesis of protein. The translation process begins at the interface of cytoplasm and endoplasmic reticulum. Okay, so that makes sense. But otherwise, how a long chain molecule like mRNA, its end will find and locate an rRNA and get aligned with it and enter into that vice? It doesn't look like a very interesting possibility from engineering sense. Okay, what is more likely is that probably it carries its own rRNA. Okay, so possibly the linked rRNA could be picked up en route from nucleolus as mRNA navigates and makes its way through pores to the endoplasmic reticulum surface. Okay, and then further question, what happens to the used up mRNA? Where does it go? Does it get dissolved in cytoplasm, or is it looped back and comes back to DNA from the other side as a closed loop for verification that it has done the job, and possibly it may initiate the rezipping process? Or does it simply enter the tank of lysozyme, the big vesicle which stores lysozyme, and gets hydrolyzed there? Okay. Which also is a possibility, of course. Now, some mutations and changes in genetic code have been studied by scientists, of course. So, chromosomes may be identified for genetic inheritance and otherwise according to two individual. So, genetic mapping is done between the offsprings, uh, siblings, and parents and child for proving that they have a heredity relation. So, yes, of course, it is used, but that is a statistical kind of a thing. Okay, matching. But the basis of these are that there are some changes in individuals. Okay, no two individual genes will be entirely, chromatids will entirely look identical. So there are some point mutations where some single amino radicals encoded or the gene may have got substituted. So on a gene, some point substitutions may be there as the process of feedback, because suppose a protein is getting formed and it finds that it is not matching with the shape of birth canal and it has to perforce bend somewhere, then it will substitute that amino radical locally there, and then somehow there is a reverse transcription translation, so the DNA knows that this change needs to be done for the game to be successful. Okay, this is a change which is required to be done because of certain changes, evolutionary changes that may have happened. So, yeah. So the amino radical point substitutions can be there, of course. Certain radicals can get substituted depending on evolution requirement. Sometimes the entire segment of DNA length can also get substituted. Sometimes a big chunk of chromosome itself may get interchanged with another one during the dynamics of cell division. Now that is called as transposition mutation. So certain part of certain chromatid gets detached from there and gets attached to some other chromatid. Okay. Now that is called as transposition mutation. It is not clear if the location of gene on karyotype really makes a difference or not. Logically, in which archive volume the pages of a gene are filed should not make any difference as long as they are retrieved and read when required and they are correlated and mapped correctly to the specific location and function of the cell. Yeah. So if it gets shifted lock, stock, and barrel, probably it may not make any difference. But if the bookmarks do not shift and only the gene shifts, probably yes, we have problems. Okay. So it is both yes and no. However, there could be some mix up of bookmark references and linkages. That's what I said. If there is a mix up of references and map, then there is a problem. So mutations must happen in both the complementary strands of DNA. If you have substitution in one in the main strand, the complementary strand also have to have the matching mutation. Otherwise, zipping will fail to happen. The re-zipping will not happen. So that's another kind of foolproofing check. So something which has to be validated for being a valid change, it has to happen on both the matching strands identically so that they remain compatible and still rezipping can happen. Now, this is a kind of per force scrutiny, and this is what provides a lot of robustness to the mechanism. Okay. Now, there is a lesser studied and lesser contemplated possibility of reverse transcription and reverse translation. Everybody is talking of forward transcription, forward translation. That okay, from DNA you have mRNA, mRNA to rRNA, then you hold on tRNA and synthesize protein according to the code that is there on DNA. But reverse can also happen. At molecular level, how does one make sure that the molecules, long chain molecules, are coming or going? It's very difficult to make out. Okay. So reverse can also very well happen through the same mechanism, through the same linkages, through the same automation that we have for forward. For example, protein can get dissolved progressively, and the amino radicals the protein has, that tRNA can come and get stuck on the strand of mRNA. So it is encoding the length of the protein, the primary structure on mRNA. Okay. And that mRNA, and of course it's matching mRNA on the upper wise, both have to be substituted accordingly, otherwise the two will not match. And then they have to go to their respective DNA, and where they do back annotation and they substitute the master in the DNA according to the mRNAs which come back feedback as feedback of evolution. Okay. From engineering sense, there has to be some mechanism of feedback if the system is so robust and if it has been improving only with passing generations. Okay. So there has to have some mechanism of reverse transcription and reverse translation of incorporating what is most survivable on DNA for progeny. Yeah. So while there is a lot of literature for forward process, the reverse has not been investigated as much. Okay. So, but from engineering possibilities, it's tempting to believe that it could be a reversible process. Why not? And there could also be reverse translation and reverse transcription from protein chain to mRNA through rRNA and tRNA up to the DNA strand. So, I mean, it is impossible for anybody to figure out with whatever molecular tools we have or methods of study we have that proteins are being formed or they are being liquidated progressively. How does one make out? Which way it is moving? It's very difficult to make out. mRNAs are coming from DNA or they are going from DNA. They are going back for the reverse annotation or they are coming forward. Very difficult for anybody to make out until one studies a molecular level movement at the single atom level. Evolution of genetic code is only possible if the inner environment of cell as existing gets encoded and substituted on the relevant mapped gene only. So just by jiggling it, some information will diffuse through at random through the mitotic spindle, through the dynamics of cell division. Some strands can get accidentally changed. No, it cannot be such an accidental process. It has to be much more regular, streamlined, proper process. Okay. So the same hardware which maps a gene to certain physical site would probably do it both ways. Could as well link the feedback to the same gene for evolution of the code. Okay. Why there would be a different mechanism? Only thing is, yes, evolution is a much slower process than the running processes of a living cell. So probably, yes, reverse transcription and translation could only be happening on cell division or with a very less statistical probability of a complete DNA strand getting substituted from cytoplasm. That is widely believed, but it is far-fetched. Okay. So probably yes, but the instances of back translation may be statistically minuscule as compared to the forward process. But it is required. Some feedback is required, some correction is required, of course. Some mechanism to link the gene to physical things in cytoplasm is required. And genetic refinements are seen to happen across several generations. Generally, I mean, it's hard to find that it happens within the lifespan of an individual cell or even individual human being. Okay. So it may require several generations for some genetic refinements to happen. Some examples like Japanese are able to make the watermelon cuboidal so that they can store it in the shelf of a refrigerator with space utilization. Okay. So what they did, they just packed these fruits while they were growing into wooden cages, into boxes. And after some generations, those plants got genetically modified. And without boxes, also, they started producing cubical fruits. Okay. Now, this is one kind, but it required several crops of doing this. Then the genetic modification happened. Of course, it would not happen in a single plant in a single generation. Okay. So, by themselves, they have become cubical. Another example is that with volcanic dust, volcanic eruptions, some part of soil goes to the uppermost layer of atmosphere, along with the bacteria that were there in the soil. So those microorganisms grow at the top layer of atmosphere, but they have over the years got genetically modified so that they have become immune to cosmic radiations. They have become immune to UV, ultraviolet, and they consume much lesser oxygen to survive. Okay. So, such slow changes through generations, such evolution is known to happen. But of course, there has to be some mechanism which connects the physical uh, atmosphere of the cell to the DNA, which is appropriate for that. So, with the correct linkage, the correct vector has to be affected. Now, the question is how is the human body encoded? In the engineering side, we are used to seeing the CAD models, which basically take tomographic sections and they make it pixel wise. So each tomographic section has pixels, rows and columns, and then you assign attributes to each pixel. however small those pixels are. And then, okay, you can have a software that can make you to visualize. You can do things like virtual dissection. Okay, so you know which layer where you go, what is the tissue like, what is the color of tissue, how does it look like. Okay, but that's only by far for visualization. Now, if you assign more attributes to pixel, for example, the properties of tissue, the hardness, the chemical level, the electrical properties, in case it is neurons. Okay, so if you want to simulate, if you want to create a working model of a human body that grows and that does movements also, it will be a humongous data which needs to be archived. Okay, so in biology, in genetic archive, of course, it's nowhere near this kind of data. Okay, so what is known about genetic archive is that it has just intelligence to selectively execute some sequences of genes for producing certain proteins and certain enzymes in certain sequence for each type of cell. Simple. So depending on where it is, which type of cell is there, what are the bookmarks, so from where to where, which sections of genes have to be read, expressed, and those have to be transcripted, translated, and rest remain dormant, and they just passed on to the daughter cells as archive. Okay, so it doesn't seem that anywhere they have the information of full body or a map of full body or pixel wise details. No, there is no such thing. Okay, now when you produce proteins at certain rate, certain proteins they carry certain shape, they take certain shape, certain size, and it leads to certain functionality of the cells, tissues, its chemical properties, its electrical, electrostatic properties, and all. Yeah. Under certain conditions, it may change to another type or structure of cell just by changing the sequence in which the proteins have to be produced. The intercellular structures and relative growth rate of cells, the cytoskeleton, and the other fibrils which are produced by the cells and which are like spanning across several cells one after another. Probably those fibrils are responsible for making the shape of organs and tissues. Okay, so they determine the ultimate shape of how the tissues should look like, how the organs should look like. Okay, and then the type of cells, of course, within the organ will determine its functionality. In the next talk, we'll be dealing in more detail about what are the specializations in various tissues and all from cell biology point of view. Then, of course, there are gross level of chemical messengers, the physiological parameters, the neurotransmitters, hormones, enzymes, cofactors, etc., growth factors in the body which control the growth rate and level of function at various tissues at macro level. Yeah. So while skeleton is majorly formed by the shape and size of body structure, it forms the shape and size of body, but it is a result of protein matrix attracting some calcium through ossification process. So the cells are secreting protein matrix, a protein fabric, so to say, which catches calcium, and the relative growth will determine the size of the bone, thickness of the bone. And bone is the next dynamic organ after the blood, say next dynamic tissue after the blood. So from inner side, it is growing. Osteocytes are the cell which are making the bone grow. Uh, Then osteoblasts are there and osteoclasts are there. So osteoclasts are dissolving it on the outer surface. So the bone keeps migrating from inner to outer surface. So it is storing calcium. It is dispensing calcium at the outer layer. The thickness of bone is storing calcium. The inner one is generating binding calcium. Okay. And of course, interior brain has bone marrow function and several biological hardware is wonderful because it is multifunctional. Everything has more than one function. Okay, but ultimately the shape of the body depends on bone. Length of bone depends on the level of growth hormone, and thickness depends on relative rate of generation and dissolution, okay, which is controlled by physiological macro parameters. It is not in genetic code, it is outside that. It's a consequence of that. How that information comes from genetic archive is a different matter. But yes, it's not clearly correlated with genetic archive. So carving of bone joint, when the bone keeps on growing and it comes to the another matching bone, now the growth terminates and it starts matching the surfaces so that you have bone carving function at the joints. So all the information is not pixel wide stored about the joints, with the geometry of the joints. Okay, they get suited upon restriction while it is in the process of assembly. Okay, so it's all the growth dynamics that determines everything. It's not so that that entire map is stored in genetics just like that. Okay, so human body structure is ultimately result of execution of genetic code in phased manner, but much more than that, it is sequence in which it happens and presence of pre-existing structures supporting conditions and surroundings at each cell, at each stage. Okay. So it's also a result of course, relative growth dynamics involving growth of various type of tissues and gross physiological parameter defining the phase of growth of tissue. Like sometimes neuron may be in adhesion phase, sometimes they are in migration phase, sometimes they are in growth phase, okay. Sometimes they are in reproduction phase, and like bones are, of course, the thickness depend on rate of forming and rate of dissolving and dissipation. Yeah. So that is the wonderful architecture of body. Statistically, the genetic code is getting refined over generations, selectively and statistically. Okay. Suppose some changes happen in the genetic archive and that improves the survivability of those who carry that modified archive. After a few generations down the line, those individuals having those changes in their genetic archive will outnumber the others. Okay. So after several generations, probably the non-optimal code will become extinct and it will diminish. Now, statistically, that is how evolution has been going forward. So, even as individuals born, live, and die, but the intelligence of the code they propagate further, that only keeps improving and it keeps evolving to become increasingly agile, increasingly robust, and increasingly refined and complicated, if you please. Yeah, there are so many checks, balances, cross checks. So substitution of half genetic material from father and mother for sexual reproduction is the fastest convergence towards positive evolution. Otherwise, you had asexual reproduction, as in grass. Life forms will grow like grass with minimal evolution. Okay, there will be hardly any improvement from generation. It will stay the same. But human beings have been a highly evolving species, and so also the mammals are. Okay, so clearly that thing is happening. With this, we have come to the end of the today's session, and I hope you have enjoyed the session. And see you in the next session. Until then, bye bye. Enjoy yourself.